Okay, this is the final, the third preparation video for lecture 19. And we're going to be looking at suppression of a mutation by a second mutation in a different gene. And this is a known phenomenon. So it happens when, let's just get the pen here. It happens when you have two genes that work together to do something. They both encode proteins, and it's very common for proteins to have to form a dimer with another protein in order to catalyze a reaction or send a signal or transcribe a gene or whatever it is that is happening. And so suppose you've got these two proteins and they interact together, and maybe they form a complex that makes a red pigment, something like that. So if you make a mutation in the gene so that the protein is the wrong shape, and now you'll see why it's important, why it's important to think about, protein a lot when we're talking about the genes, because that's really where the phenotype is set up. If the shape of the protein changes, now it can't bind with the other component. And so the two of them, the organism is wild type for this gene, but because this one is mutated, they can't catalyze that reaction. So you're going to get red flowers in the case of being wild type for both, but you're going to have white flowers here because that red pigment can't be produced. But suppose there was a mutation in the second gene now, and by chance that mutation basically changed the shape of that protein, so now it actually could bind to the mutant one. So we call that a suppressor mutation because the change in this one overcomes the problem with this one. And it's happened by a sort of, you'd almost call it like a complementary mutation. Suppose there's an arginine that's supposed to be right here and it binds to a histidine or something like that. And now you've got a different couple of amino acids, glycine or something, and it cannot bind to this pocket where the histidine is. But if you put in a amino acid that's a little bit of a different shape, now they can bind again and you restore the phenotype. So now you're back to having red flowers again. But what that means too is, like, suppose you do a set of crosses and you manage to isolate some plants that are homozygous for the mutation in the suppressor gene, it now has this wrong shape as well. And so if you're mutant for this one, but not this one, then you're going to still have white flowers. So think about a dihybrid cross. Think about, M plus M, S plus S crossed to, it's a plant, so you could self-fertilize it. What would the ratio that be that you would expect? You'd expect that 9 to 3 to 3 to 1. And what color would each of those categories be? And I'll be asking you questions about this in the class. Oh, and in fact, we'll probably go through this and fill this stuff in the class. So I'll have this for you to do on your own, but then we'll probably have like a quicker question or two for you to answer. Now this is a different kind of suppressor, and this is from flies. This is an organism that I know more about. And it turns out that there are some genes that when they are mutated, they don't have any phenotype by themselves. They are only recognizable based on how they interact with some other gene, either wild type or mutant. And so this is an example. So there's a gene, I keep calling it, it's actually called purpuloid because there's a different gene called purple and this one's slightly different, but I will probably just keep calling it purple. And basically, instead of the eyes being that sort of normal brick red color, you've got this kind of purplish pinky kind of color. It almost looks brown under the microscope, but depending on the lighting, you know, how intense the light is and so on, it's called purpuloid. And so suppose we cross flies that are homozygous, the pure breeding for both purple and the suppressor gene, and then we have these other flies that are pure breeding for the suppressor gene that has no phenotype by itself, and homozygous for the wild type of purpuloid, and so now they also have wild type eye color. But when we make the dihybrids, and we can't self them, but we can cross them to siblings that have the same genotype from the same cross. And we get, of course, 9/16 that are wild type for both, 3/16 that are wild type for the purple but mutant for the suppressor. So these are also going to be wild type. 3/16 that are mutant for the purple and wild type for the suppressor, so they're going to have the purple eyes. And then 1/16 that's mutant for both genes, and that's where this mutation suppresses that mutation and returns the eyes to the wild type color. And you know, I think they do know how this works now. They didn't for years and years, but I am not aware of how it actually works, because it's a really strange one. And so this is a characteristic of a recessive suppressor that's acting on a recessive mutation that does not have any phenotype of its own, right? Identified only, you can only see the effect of it when it's in combination with the mutant. So this is the only place you can see the effect of the suppressor mutation, even though, you know, 3/16 of the progeny are mutant for it, but they also already have the wild type purpuloid gene. Okay, so hopefully that's helpful. Those are just two examples of different kinds of interactions. So the kind of question to be thinking about for those is, you would probably be given a scenario. Here's this gene with this phenotype, and here's this other gene, and it's mutated this way, and this is how it suppresses. Now tell me what the phenotype will be and explain why, you know, in a dihybrid cross. Or here's a, you know, a dihybrid cross, here's the results, you know, tell me which kind of suppression is this. Is it like the first kind, or is it like the second kind, where the suppressor itself doesn't have its own phenotype? And you can tell that from the ratios, and then just be prepared to kind of explain in your own words, you know, how that works. Now there's some other things. This takes us back to the idea of hypomorphs. So you might remember right at near the beginning when we're talking about different kinds of mutations and stuff. I talked about hypomorphs and amorphs and hyper and neomorphs. And hypomorphs are ones that have less gene function than normal, or less protein function, I should say, but they're not completely null. They're not amorphs. Amorphs are when either the protein's not being made at all, or it's a completely useless protein that can't do anything. Okay, so sometimes you will have, you know, say two proteins that have to work together. They're transcription factors, so they have to bind to each other, and they have to bind to the DNA. Okay. And when you make a mutant in one or the other, they are still weakly active, active enough that you have a normal phenotype. But when they're both mutated, you have a more severe phenotype. So we'll think about this situation where we've got a transcription factor. Each of them binds part of the sequence that they're targeting in order to initiate transcription of the gene, and they have to be bound to each other in order to do their job. Okay, now here's a mutant in gene A. It can still bind to B, but it doesn't bind very well to the DNA. But because it can still bind to B, and B is all right, it's kind of anchored in the right place, and this allows for transcription. So there's enough enzyme function for the organism to survive. And the same with B. You know, if we have a mutation in gene B and the protein can just, you know, kind of bind a little bit to the DNA, it's not really very good, but it's attached it's been bound, dimerized with gene A, and the gene A protein is nicely bound to the DNA. So in both of these cases, you have enough enzyme for survival. But if both of them are mutated, there they are stuck to each other, but neither one of them is able to bind nicely to the DNA because they just don't have enough of the correct, shape, whatever the protein is supposed to be shaped here to recognize the DNA sequence. So you've got your dimer like floating off and not transcribing the gene. And this would not be enough for survival. And we call this synthetic lethality. Neither of those two mutations by themselves is lethal. They both can hang on and survive, but when you make mutants in both of them, it's lethal. And that's a characteristic when genes are acting together in some process. And that's, there's a large-scale project in yeast, in fact, where they're looking at, they're making every possible combination of mutants in the yeast to find out which ones are synthetic lethals, because that suggests they act together in the same pathway, and then they investigate further and find out how exactly they work together and what they're doing. And it's a really large-scale project, so it's really interesting. Okay, now the last little bit is just a few samples that I've copied from different books and so on, and they just kind of show you the different kinds of interactions. There's cases here with the 4 distinct phenotypes, some of the eye color that I did, and the snake example, and a couple of others would fall into this category. Complementary gene action, the eye color ones with the cinnabar and the scarlet. Recessive suppression, there's different ones of that. The epistasis with the flowers, which actually we can leave this out, but the recessive epistasis that we looked at, and dominant epistasis, and then duplicate genes. So these are the most important ones. And this shows you what the ratio is. And this is helpful because it just shows you which categories are grouped together in the different kinds of ratios. And then these are just a few other examples, and we'll probably touch on, we'll touch on this one for sure in lecture 20. This is the squash example we looked at. This is an interesting one. You've got here a 9:7 ratio, so you might want to consider how that acts. Would be a good question to ask. Squash color we looked at. You can think about this one if you want to. It's an interesting one. If you can think of a way that it might work with a 13:3 kind of ratio. The shepherd's purse. And I would probably suggest that you leave this one out. I included both of them because they just have different ways of showing you kind of how, because you know what the genotypes are going to be, right, for each of these. And if you know that, the genotype, and you see the modified ratio, you can figure out which of the categories are each genotype, and that helps you sort out what they're probably doing. So you only need to be familiar with the ratios discussed in the lecture notes and that we cover in the class, but there are other ones that are potentially of interest, and you could see something like this on an exam, with some sort of explanation. And if you've thought about it in advance, of course, you're better prepared to answer a question like that. I guess I was showing them. So the way, when we're thinking about gene interaction problems, the first thing you want to do is determine whether you have 1 or 2 genes that are responsible, right? If a complementation test is possible, or if you've got information about that, then you can determine 1 gene or 2 genes. Or you should be given data about crossing F1s to each other, and then you would look for the kinds of ratios that you would expect if there's 1 gene or 2 genes, right? And once you have something that's, obviously a modified dihybrid ratio, or obviously, a 1:2:1, then you can decide what kind of ratio you're looking at. And then once you've done that, if you've decided that it's a modified dihybrid ratio, then you just write out the genotypes for the, the 4 different categories, and then figure out, like if I'm seeing 9 to 7, then I know that this is all one phenotype and this is all another phenotype. And that suggests that, a homozygous mutant for either of the genes is giving you the same phenotype, because, and you could make a reason for that. Or a 9 to 3 to 4, in our case with the flower color and stuff, you can explain how if you're mutant for this gene, it acts before this gene's product does. And so if you're homozygous mutant for this, it doesn't matter what genotype you have here, because the whole pathway stops at this point. And you can do the same for all of these. Just see how they're grouped in a way that makes sense, and then figure out how that works. And then with the suppressor of purple, there is the 13 to 3 ratio as well, which is sort of a combination of that and that. It's when, the double mutant suppresses the phenotype, and the wild type is wild type, and then the other two are mutant. Okay, so I think that's everything. We are going to start the next lecture. It's just, it'll be fairly short, and there'll just be lots of clicker questions. I'll explain how a lot of the mammalian coat color genes work, and then we'll talk about, I'll ask you questions about, here's a picture of a cat, tell me what his genotype is for this gene or that gene, just so they have lots and lots of practice at doing that sort of thing to help prepare you for any potential exam questions. Our exam is still a couple weeks away, but it's not bad to start, thinking about it.